Okay, so when we talk about cells dividing, you have to first talk about DNA replication. So, little quick review about DNA replication. Hopefully, you have already studied this with your lecture class, because remember, lab and lecture go together. This is the picture that actually comes from our lecture lecture book, and DNA has to replicate perfectly before a cell can divide. And so when we look at any cell in our body, any cell that's out there, cells are always somewhere in the cell cycle. So cells live in a cycle. And when we look at the cell cycle, it depends on what kind of cell we're looking at, where that cell actually is. So if we look at many of the cells in our body are actually in what we call the G0 stage or G0. I mean, in South Africa, we would say naught instead of zero. So that's kind of why I say G0. But these are going to be cells that are not dividing. And those cells may never ever divide again unless something happens to them and they need to be replaced. Or maybe they're a cell that divided many times until you grew to an adult and now you don't grow, so you're not going to divide again. But things like when you damage or you injure your body in some way, then the cell automatically gets signals to repair itself or repair the cells around it. So if you skin your knee, you've got to replace those. If you break a bone, you produce new you produce new bone cells. And we're even discovering, they used to say, you're born with all your brain cells, but we're also discovering that even brain cells can regenerate. So for the most of the time, so there's those cells that are not dividing, but then there are other cells that are somewhere in the cell cycle. And the amount of time they spend in the cell cycle is going to depend what kind of cell they are. So the two main phases. So if I start at the end, at where G0 starts, and I go all the way around here, that phase is called interphase. And then the other part of the cell cycle is this M phase, which is mitosis. And mitosis ends with a division into two new identical daughter cells. And then the fate of those daughter cells is going to depend on where they are and what needs to happen to them. When we look at interphase is divided into these three parts. So I see in this picture, the interphase is actually showing on the inside, but people kind of miss that. So here's our interphase. All of that is interphase. So when we look at interphase in the cell cycle, G1 is the phase where you have growth, because if that cell is just newly divided, it needs to be bigger. It needs to actually grow to a certain size, because you can't just take a cell and keep dividing it in half and in half again and in half again, because the cell would just be too small. So a cell has to be a certain size before it can even undergo cell division. And then there has to be preparation for DNA synthesis. So before the cell's going to divide, you have to have all the right things to undergo DNA replication. So proteins. So if you think about all the different enzymes that are required for DNA replication, those have to be made and prepared before the cell can even go into DNA replication. The other thing is we have specific checkpoints. So there are checkpoints to prevent cells from going into the second part of interphase, because if the cell's not ready and it carries on, it's not going to do what it's supposed to do, and ultimately you're going to have a mess and the cell's going to have to die. So G1 is your preparation for DNA synthesis. S phase is where you replicate all of your DNA. Once you finish replication, there's going to be a further stage of growth. We call that the G2 phase. And then at the end of G2, if everything is okay, so we're going to have another big checkpoint over here, if everything's okay, then the cell's going to commit to go through the division phases known as mitosis, where you have chromosomes and those chromosomes separate in to two identical daughter cells. So depending on which book you look at, which picture you put at, there is a phase that sometimes is not discussed, and that's called prometaphase. And prometaphase is just the phase between prophase and metaphase, because when you look at a cell in prophase, it's very distinctly different from a cell in metaphase. And prometaphase is where you go between. It's basically, how did you move from prophase into metaphase? And we'll discuss that in further detail. So what I quickly want to do is just go through DNA replication. You probably maybe saw it in your lecture class, but this can be a review. And then just go through what we would have done in our lab activity. So I'll just go through the pictures that we would have taken. So some of you maybe, when things were still crazy, go, what were these images I was supposed to post? Well, the images would have been, if we did this lab in class, these would have been the images you would have taken on your table and then posted them for your group to have a record of. So this next picture here is talking about all the proteins that are involved in DNA replication. So you can think about these are going to be made in G1 phase. These are the proteins that need to be made. So remember, to make a protein, a gene has to be expressed. The messenger RNA has to go out of the nuclear pore into the cytoplasm, and even in the cytoplasm, it has to be translated into an actual protein. That protein has to fold in the perfect shape. And now any of these proteins that are part of DNA replication, they have to go back through the nuclear pore and go back into the nucleus, because DNA replication occurs inside of the nucleus. So when you think of that nuclear pore, think how busy it is as a channel. Things are going out and things are coming in, but only very selective things. So proteins that are necessary for DNA replication have to get back into the nucleus. Okay, so let's look at this picture here. If we were doing this lab, you would have had some cardboard pieces of DNA, and one would be, this is basically your original DNA strand. So we had color coded it as pink and blue just to help showing separation, nothing to do with being male or female. But what you would look at is, let's try and see orientation of these strands. I'm going to see if I can just zoom in on my picture a little bit, move it over. So how do you identify orientation of these strands? So we're going to look at the P, the phosphate in the backbone, and we can look at the P on the pink strand. And if you look at this phosphate, the phosphate is always your 5', and then this part here is your 3'. 5', 3'. And if you actually look at the sugar, the piece on the sugar is pointed in one way on this strand. So it's your sugar that determines where your phosphate's attached. Your base is always on carbon number 1. And now we're looking at our other strand, and there's our phosphate. So there's our 5', there's our 3' end, there's our phosphates. And so you can see also from just the direction of the sugar that this strand is going in the opposite direction. So when we look at our basic strand of DNA, it runs anti-parallel in opposite direction. And that's really important, because when you do DNA replication, DNA polymerase, the enzyme that adds nucleotides one base pair at a time, it can only work in one direction. Okay, so here's the picture that you would have seen in your lab, in your lecture video. So we have, firstly, we have an enzyme known as helicase, and helicase separates the strands. So helicase is making those double bonds into single bonds. And then you have some single-stranded binding proteins that attach to them to prevent the single strands from going back together. Then the next thing that has to happen is we have an enzyme called primase. And what primase does is it starts making the template strand double-stranded so that DNA polymerase can now work. Because DNA polymerase always needs a 3' hydroxyl group to bring the next nucleotide on. So you can see in these two strands, we're going to replicate in opposite directions. So what we would have done in our lab is we would have actually had a mock copy of helicase, and now you can see how I've opened up my parental strands. If we look at this strand is going in 5' to 3' direction in that way, and then this one is going in the opposite direction. So you can see the, whoops, let me just redo that, it's the other way around. So this is starting, this is 5' end, going 3' that way, and then this one is 5' going 3' in this way. So you can see the helicase is opened up. The next picture we would have done is we would have put our little fake single-stranded binding proteins down to keep the strands apart so that that G doesn't magically go and repair with the C. Okay, the next step, if we go back here, is I need to put primers down. Now, our primers are primers of RNA. So if we had done the lab, our RNA nucleotides were pink, and how we can distinguish an RNA nucleotide is that RNA has U's. So you can see there's a C going to pair with a G, and there's a U. And what you're seeing in this picture is that we put in primers in different places. So one set of primers is being put in quite close to where the helicase opened up. So we got one set of primers. On this strand, we add in the primers up here. And on the other strand, you're starting way further back, because it's pointless to put primers right next to the replication fork because you're going to follow the replication fork. So on the other strand, you would have put your primers where it first opened up, because that's where you can do it. Complementary base pairing. You can see the complementary base pairing. Now that you've got your primers down, now we can bring DNA polymerase in. So DNA polymerase adds nucleotides, one nucleotide at a time. And you can see on my top strand here, once I have my primers down, I'm going to keep adding them. And this DNA polymerase is basically following the direction of the helicase. So we can just keep going. The other strand, we put our primers down. So this is at our replication fork. We put our primers down, and now we're going in the opposite direction. So one of the things I can just really encourage you to make a note of as many times as you can is that DNA polymerase always works in the 5' to 3' direction. So regardless on which strand I'm working on, I'm still going in the 5' to 3' direction. So here's a picture of students in class, and you can see here's this strand. There's my little pieces of RNA, and now the DNA polymerase has gone in, and this strand has been adding one nucleotide in that direction. And you can see on the other strand, my DNA nucleotides are being added in this direction. So my DNA polymerase is working in opposite directions. This side is going, let me change my pen color so you can see a little bit better. This one is going to keep going towards the replication fork. This strand is going away from the replication fork. Okay, any questions on that? What I've explained so far? We have a chat message. Cheyenne, we're not submitting this lab report. There's going to be some other 2 reports that I'll be showing you a little later that we're going to do on mitosis and meiosis. But what I'm first doing is reviewing DNA replication that we would have done in class. Okay, so let's keep moving. So we're making two complementary strands and the DNA polymerase is working in opposite directions, because DNA polymerase can only work in the 5' to 3' direction. So what would happen is you would end up with both your strands being made, but what would happen is one of them would be able to go continuously, and as we kept opening up the strand at the replication fork, so this strand here, this helicase, we would keep going and opening up, and that strand would able to just keep going towards the end. So hang on a sec, let me just take the picture. So what we would be doing is we'd be keep copying on the blue strand. But what would happen is this pink strand, when this opened up here, we would need to start again with RNA primers. We would need to start putting new RNA primers and then come in this direction again. So when you are making DNA, when you're replicating, one of your strands, the strand that is made continuously, that follows the helicase and can keep going and going, is known as your leading strand. The other side is going to keep having to start in again. It's going to first have to put some RNA down, and then DNA polymerase can come in and make some more pieces. And then ultimately it's going to bump into the piece that was already made. So if we go back and look at this picture, you can see that we've made this strand starting further down from the replication, where we're going in this direction. So we're going to need to put some more RNA nucleotides down, and then we're going to make a piece, and we're ultimately going to bump into the piece that was already made. So this side here is going to be considered lagging. And this one side over here is going to be considered the leading strand. So leading follows, opening up, keep going. Lagging, have to keep putting RNA nucleotides down with primase, and then you keep bumping in. So the last thing that happens is we have to remove our RNA out of our new strand that we've made. So we need to come in and take out these little pieces of RNA, and then we need to put more DNA nucleotides in. And ultimately what ends up happening is you end up with these little gaps in the backbone. And these little gaps can't automatically just be filled in when DNA polymerase comes down. DNA polymerase can't close that gap. So it can put new nucleotides down, but it can't close that gap. It doesn't work for the way the enzyme works. So think of the shape of an enzyme. An enzyme shape has to fit perfectly to the shape of the reaction it's catalyzing. And when you have some DNA already down, the DNA polymerase, the shape's not right for it to make that phosphodiester bond between the sugar and the phosphate. So in order to close that gap, you're going to need a special enzyme to come in, and that enzyme is going to be our enzyme ligase. So ligase is the fifth protein that you need to come and close that gap in the DNA backbone. So what's really cool about this experiment, when we just did word replication and maybe we replicated maybe 50 base pairs, what you end up seeing, let me just pull my picture down a little bit more, but what you end up seeing is at the ends, what we would have had at the ends of both of our chromosomes, we would have ended up with a piece that was missing. Because we can't add a DNA polymerase down at this side because we need double-strandedness. So you always end up with a few bases at the very end of your DNA sequence that can't be filled in. So let me see if I can pull this picture all the way down to the other side. Oh, I don't have a picture of the other side, but it would have been the same situation on the other strand. So when we're making our new DNA strands, one of the strands will always be missing a few sequences on this side, and the other new strand will always be missing a few pieces on the other side. So what does this mean for our cells is in our human chromosomes, we have 23 pairs of chromosomes. So think of them as a linear piece of DNA. And what happens is the ends get shorter and shorter. So we actually protect the ends of our chromosomes with, they're called telomeres, and telomeres are basically a cap, and it's a repetitive sequence. So it doesn't matter that we keep not replicating the very, very tips. But what happens is over time, the telomeres shorten. And once the telomeres are completely gone, the cells naturally die. So we know that most of our cells can divide for about 50 times, and then they can no longer divide. However, in cells that need to divide very rapidly, very early on in our lives, when we were a zygote and becoming a fetus and growing to a baby by 9 months, incredible growth, certain stem cells have an enzyme known as telomerase. And what telomerase does is it helps repair those ends. So it keeps adding some repetitive sequence at the end of our chromosome tips so that they don't shorten. The other thing is we have telomeres at the end of both of our cells. So this shortening is happening on both ends of our chromosomes. So just a quick review, when we think about a chromosome, the actual structure of a chromosome, we kind of like to draw them as X's, but that's just a real sort of representation of what they look like. They don't exactly look like that, but chromosomes have different parts, and the part that holds them together is known as the centromere. And then the part that is above the centromere, which is the shorter side, is called the P arm. So these will both be P arms. And then the part that is longer is known as the Q arm. So this side is going to be the Q arm. And when chromosomes condense down, your genes are always in the same place on the chromosome. So you could have a gene on the short arm of your chromosome that when that DNA condenses down into chromosome, that gene is always in that same spot.
Does that go for all chromosomes?
Yes.
So like only one part of the entire chromosome holds the genes. What's the rest of it then?
No. Your genes are up and down your whole chromosome. So depending on which chromosome you are, you maybe have 1,000 genes on them. If you are a short, very small chromosome, you maybe only have 200 genes on you. But we have altogether, humans have about 25,000 genes that have a specific address on a specific chromosome. So insulin is always on the short arm of chromosome 11. Beta hemoglobin, a gene that we talked about a lot, beta hemoglobin also happens to be on the short arm of chromosome 11.
Oh, okay. So each gene has one specific location. Okay.
Yeah. So if you think of, you have that long piece of DNA where the address of where your gene happens to be when it's just chromatin. So say this is your gene for insulin. And when that long piece of DNA condenses down to form a chromosome, that's where your insulin gene is. And remember, your DNA, when you see chromosomes, the DNA is already replicated. So you've got 2 copies of your insulin gene on one of your chromosomes 11, and then you have an identical copy of chromosome 11, because we always have 2 copies of every one of our chromosomes. So another gene you'll get to hear a lot about is the gene for sex determination, and that gene is called SRY, and that gene is found on the Y chromosome. And that gene is one of the most important genes in determining that the fetus becomes a male. That gene sets in motion the cells to turn into a testis and make a male rather than an ovary for a female. So just to go back to what I was telling you, how chromosomes shorten over time, this is a picture that I found on Wikipedia that kind of homes in on it, showing that tip on the end that's not being replicated. If you have telomerase, what telomerase can do is kind of just keep extending it so that you don't fray down. But telomerase is not present in all of our cells. But if we look at some cancer cells, there is cancer cells that may have overactive telomerase, and that's why cancer cells can keep replicating and end up forming a tumor. And then what about people who have problems with their cell cycle and cell division? And you've probably heard of these people. It's a condition known as Werner syndrome. And in Werner syndrome, these individuals age incredibly rapidly. So these children that you're looking at look like old men or an old lady. And what's happened is they maybe live for 20 years, but they age from, they age 5 times as fast as anybody else in the population. And it's due to something wrong with their genes that help with cell division. So their cells die, instead of after 50 cell divisions, their cells maybe die after 10. And so we know cell division is related to aging. In fact, another story that I'll just add here is telomeres. So our telomeres are a certain length. And when we look at males and females, our telomeres are the same length until the age of 35. And if we look at telomeres in females versus males after 35, on average, females' telomeres are still longer than males. And one of the things that we know about aging in populations is that males don't age as well as females. If you work in any of these assisted livings or retirement homes, the proportion of males to females is very different. There's usually a lot more females than males. So we do know that your telomeres are also something to do with longevity. And we also know that stress messes up your telomeres. So look after your telomeres. Try stay as healthy as possible. Don't damage your telomeres. Don't smoke. Smoking damages telomeres as well. And now you see, this is something you can maybe talk to your family about, is look after your telomeres, because if your telomeres fray too early, your cells are going to die earlier than they should have. Okay, let's move into a quick discussion about mitosis and meiosis, and then I will show you the assignments that you have to do, and then you will be free to go off and do them, and I'll show you how to do them first. Okay, so here's your basic structure of a chromosome. Now, the only time you see chromosomes inside a cell is if that cell is undergoing division. So the only time you're going to see the structure as a chromosome consisting of 2 identical sister chromatids is if that cell is already in prophase of cell division. Up until prophase, when you look in a cell, the DNA is still chromatin, even though it may already have been replicated in the S phase of cell division. So again, structure of chromosomes, it's always important to know them. When you're looking at a chromosome, you have 2 identical sister chromatids. One is an exact copy of the other. You have the shorter side is known as the P arm, which is above the centromere, and then the longer side is the Q arm. So this is Q, this is also Q. That's P, that is also P. And they're made up of that long DNA molecule wrapped around proteins called histones in order to condense down into these very distinct structures. Okay, so what is mitosis? So once we have gone through G1S, G2 of mitosis, interphase, the cell now commits to undergo mitosis. And mitosis is made up of these distinct stages. If you just remember an acronym PMAT, that will help you remember what's happening, the order that they go in. But interphase is not part of mitosis. Interphase is the stage of a cell before you commit to mitosis. And it's the stage that a cell goes into the minute mitosis has ended. So when we look at interphase, prophase is where you see appearance of chromosomes. Metaphase, those chromosomes are lined up in the middle of the cell. And you can see there are these specific little structures that are being formed that attach to our chromosomes to ultimately pull apart the sister chromatids. So just when you're looking at a chromosome, it kind of makes sense if they're exact copies that mitosis is going to pull that side away from that side so that each new cell gets an exact copy of all of the genes that are up and down each of those chromosomes. And the part that's important in mitosis is actually at the centromere, because there's these special structures known as spindle fibers that are made by a structure known as a centriole. And spindle fibers are microtubules. So jump back into chapter 3 and review microtubules, which are little pieces of protein that assemble together in a hollow-like tube structure, and they attach at the centromere and pull them apart. So you can see what's happening in metaphase. Here comes our spindle fibers that are extending from opposite sides of the cell. We call them the poles. And they're going to attach at the centromere. And now when we move into anaphase, those spindle fibers are pulling the chromosomes apart, pulling them to opposite sides of the cell. And then we move into telophase. Oops, I have to take out my. When we move into telophase, you now see a nuclear membrane reforming. So in prophase, there's kind of still a nuclear membrane, and that nuclear membrane dissolves between prophase and metaphase. So in prometaphase is where the nuclear membrane goes away. In telophase, you're going to reform that nuclear membrane, but you can see that the new cell has a blue and a pink copy of a chromosome, and then you're going to have a final, the last part is where we separate out, we divide the cytoplasm, and the cell membrane pinches together, and you end up with two identical cells. So diagrams like this are very, very simple versions of mitosis. But if you think about your own cells, you have 23 pairs of chromosomes. So you actually have 46 chromosomes all lined up in the middle, and then each of them pulls apart. So can you imagine the mess inside a cell if it wasn't highly, highly organized? So what we're going to do in this lab is we're actually going to look at onion cells that are undergoing mitosis. And so in the worksheet that's on Canvas that I'll pull up and show you, you're going to link to this website. It's the Biology Arizona Edu Cell. And you're going to actually go through identifying cells in different stages of mitosis and then count them, and you basically be putting them into little boxes, and then you'll take a screenshot when you're done. So when you look at real live cells, all of a sudden it's not as neat as the little little drawings that we give. Real cells look a little bit more complicated, look a little bit more messy. But in looking at these pictures here, you can actually see what's happening in each of these cells. So first stage is prophase. So how can we identify a prophase cell? Well, a prophase cell, you can still see the nuclear membrane. You can still sort of see it happening in one space, but you already see thickening. So you kind of see the little appearance of almost like little sausages happening inside that cell. So prophase, they're still compact in the middle of the cell. This phase here is metaphase. And you can see metaphase doesn't look anything like when we show you, oh, look how beautifully they're lined up down in the middle. You know, look how nice they look over here. When you look at real life metaphase cells, they're kind of all in the middle, but remember those chromosomes may be sticking up or sticking down, but you can see them distinctly stuck together. So M for middle, M for metaphase. This next phase here is anaphase. And you can see how they are making that almost little V-shaped pull to the other side. And then this last stage here is telophase. And so you can see in that almost same amount of space, you're now seeing two new nuclei being formed, and the chromosomes are getting less dense. So they're not as tightly packed as you start to see the formation in this picture, versus they start to decondense, if that's a word, become less relaxed. Now, when you're looking at onion cells, what is something we have to remember? The difference between an animal cell versus a plant cell? Someone can add in and give me an answer. What's one of the distinguishing features between an animal cell and a plant cell?
Cell wall.
Exactly. Do animal cells have a cell wall? No. So they have no cell wall, they have a cell membrane. Do plants have a cell membrane? Please say yes. So plant cells have a cell membrane because all living organisms have a cell membrane. But in addition to that, they have a cell wall. So think of a membrane as being soft, and that you can actually push that membrane down and divide into two cells. Plant cells, not so easy. What a plant cell has to do is it gets little vesicles deposited down the middle, and inside those vesicles, what makes a cell wall? Cellulose. And those little vesicles of cellulose get deposited and eventually fuse together. So we build that cell wall back up. And so when you're looking at plant cells, when they're undergoing mitosis, so this will be a cell that's, say, in prophase, when the cell size is about the same, but you start to see those two nuclei forming. So a little bit more tricky in plant cells because you have to grow a cell plate. So that's called a cell plate. And it consists of vesicles of cellulose that actually help it form. So if you are looking down a microscope, which is what we would have done in class today, we would have looked at root tips of onions. So if you were looking at an onion, you would look at roots, because roots have an actively dividing region. And then roots also have what's known as a root cap to protect the actively dividing region. So we would have taken cells of onion root tips, and then we would have focused up on the microscope 40 times magnification at that spot. And then you in your groups would have then counted cells and then recorded which ones are in prophase, which are in metaphase, which are in telophase, and which are in interphase. So interphase is going to be the cells that are not dividing. So if we had to look at the circle here, which are the ones that just jump out of you immediately as being, oh, I know exactly what that one is. Hopefully you would say, oh, that's a very distinct picture in there. Anyone want to give a guess what those are in?
Those are metaphase.
Definitely metaphase. But you see how it's not neat and tidy like we like to draw our chromosomes in our little simulations, draw them nice and neat. They're actually held together in metaphase, and metaphase is actually a long period, because what has to happen is all the chromosomes need to get to the middle before there's the go-ahead to separate out. So there's actually another checkpoint in metaphase in cell division to actually make sure all the chromosomes in the middle before they're allowed to separate out. Let's see, what about this? If we look at this cell over here, what would you guess that one is in?
Telophase.
So not really anaphase, it's kind of just past anaphase, because you're starting to see it's kind of more condensing at the end. So that one, this one here would probably be telophase. And what will happen is you'll get to practice, and if you make a mistake in the simulation that you do, if you make a mistake, it'll say no, try again, and it'll tell you why. So the ones that get hard are to distinguish whether they're in interphase or prophase. And then you'll get good at looking at that. So if we had to look at this cell versus this cell, if you're looking at that cell,
one, it looks like
interphase.
That one would be in interphase. And then this one here, you can start to see appearance of chromosomes. So the minute you see going from chromatin to chromosomes, then this one here would be prophase. So how about that one?
Interphase?
Interphase or prophase. So again, it becomes a little bit more subjective, but if you look closer at that one, you can see there's distinct lines between the DNA. So that one was probably prophase. But again, you start to see it's a little bit tricky. The other ones, if you look at, say, this one here, let me see if I can zoom in. I have to first, let me just take all my other. Let's zoom in on this cell over here. When I zoom, my markings don't zoom. I'm going to zoom in the background. When you zoom in on this one here, you actually see a dark spot still in it. Anyone remember what the dark spot inside the chromatin often represents?
The nucleus.
So the whole thing, this whole thing is called the nucleus, but the dark spot inside, nucleolus, which remember codes for your subunits of your ribosomes, ribosomal subunits. So when you're looking at them and you can see distinctly that dark spot, then it's more likely going to be interphase. Ultimately, in the end, it's not going to really matter, because when you do the practice assignment, it's going to give you lots of opportunities to make mistakes, and ultimately you will get your count right, because it won't let you put the incorrect count in. So in the spread, I'm not actually seeing a nice anaphase for us to look at. So if we go back to this picture here, when you're looking at anaphase, you can kind of see that almost V shape of the chromosomes pulling apart. Now here's something that's really cool, is before people knew DNA was the genetic material, they had already seen chromosomes behaving in mitosis in cells that they were looking at. But they didn't know that it was DNA. They didn't know that DNA was the genetic material, because chromosomes are basically, when you analyze them, they're DNA and protein. And so it took until the 1950s for us to say, well, it's actually the DNA part of that chromosome and not the proteins that we know is the genetic material. Okay, so let me take away my iPad and open up your assignment that you're going to do on Canvas. So what you want to do for your lab is go to the modules, and the one that's then going to show up for you this week is lab 12, mitosis and meiosis. Next week's lab's still not ready, so don't worry about that one. Let me just move you guys away. So what you can see is in this module, the first one is DNA replication videos to watch and help you understand the process. This is the Howard Hughes Medical Institute DNA replication showing, and it's an animation showing all the different parts. And then this is a video that I made that's actually on YouTube where I explain DNA replication using the smartboard. So this is another one if you want to, a review of all the different enzymes and how the details of it works. Then you can click next. Let me click next. The next part of that module is two really good mitosis and meiosis videos to watch. This is Paul Anderson. He is the AP Bio hero. He made AP Bio videos probably 5 or 6 years ago, and he's saving every teacher in America's life because he's got such good videos showing processes. So watch the mitosis video, do the mitosis worksheet, and then come back and watch meiosis. Don't watch mitosis and meiosis before you do the assignment because you might get confused. And we want to try to keep you not confused. So once you've watched your mitosis video, this is the instructions that you're going to do. So here's the link you're going to go to University of Arizona. And before you just click on that link, first read the directions and print off this mitosis lab report document. Because what you're going to do is answer some questions on that document, and then ultimately you're going to take a screenshot of the activity that you do. So in the University of Arizona onion root tip, what it's going to do is walk you through identifying each phase. So what you ultimately will do is click on which phase you've seen. So if you saw that, you would say, well, that's metaphase. You'll click on metaphase, and if you're correct, then it'll put it in a tally. So ultimately, you're going to end up with a picture of your results, and you will count each cell in each phase and record that on your worksheet. Here's just some other little pictures to help you as a guide in identifying. So we've kind of talked a little bit about this already. How do you know the difference, whether it's interphase or prophase? And again, these kind of between interphase and prophase gets a little bit sticky, but you'll figure it out. It's not hard. When you're finished, you're going to need to take a screenshot of your completed table, and then that's going to get included with your mitosis lab report document. So this document you can fill in online. If you print it off and fill it in, then you can upload it as a PDF file into this assignment, but you'll see there's some definitions you have to write about. You also summarize in your own words what's happening, and here you're going to record your counts, and here you'll insert your screenshot, and then you'll upload it to Canvas in this spot. So you'll see at the bottom here, it'll say upload it as a file doc. Try and, your screenshot, if you just do it as a JPEG, make sure it's actually easier if you embed it into the Word doc, because then it shows all as one file. students often, if you upload more than one thing, then your second file doesn't show up because you knock out your first file. But if you download this document and then take a screenshot and incorporate it in, and just as a reminder, everybody has free access to Office 365. So getting access to Word is not a problem. Just you get free access if you're a student at FSW. Okay. Anybody have any questions about this part here that you need to do? Okay, so good review for this. Okay, then the second activity is on meiosis. So the meiosis lab report, again, also has a document for you to fill out. The activity for meiosis is you're going to go to concord.org. And in this one, you're actually going to do meiosis of, you're going to make babies with a dragon. And what you're going to do is look at what the offspring end up looking like. So let's just talk a little bit about meiosis. So let me just pull up another folder.
Donkey's back at it again.
What's that? Sorry, Noah, what'd you say?
The donkey's back at it again. Shrek joke, sorry.
Okay. Okay, so let's have a look at, so if we were, let me just share my screen, sorry. In our class, when we did meiosis, we actually had, oops, I didn't screen share. Screen mirroring. Okay. So had we been in class, and this is also one of my favorite labs to do with folks, what we would have done is I would have given you, everybody would have got a set of chromosomes of a bird. And then what you would have done is in your chromosome sets, there would have been 2 colors, and then this would be the parental cell. So this would represent you. And what you would see is that when we look at the chromosomes, there's 6 chromosomes in this organism. And anyone want to guess whether it's a male or a female?
Female.
Why is it a female?
Because there's no Y chromosome.
Right. So when you're looking at this, you're actually seeing there are 2 X chromosomes. So what's really nice about this lab is when you're looking at your chromosomes, the blue came from dad, so that's your paternal X, and that's your maternal X. So this individual would be a female. And then if we look at the other 2 chromosomes, we have a long chromosome from mom, we have a long chromosome from dad. And if you look up and down the chromosomes, what's nice is you actually see the genes are represented by letters and they're in the same place, okay? And then you have a short chromosome from mom and a short chromosome from dad. So what happens is this would be in a parental cell that undergoes meiosis. So there's very few, there's only, there's special cells that undergo meiosis, and those are called germline cells. And if you are a female, your germ cells are your ovaries. And if you are a male, they're your testes. And of course, we're using plural. You have 2 ovaries and males have 2 testes. So it's plural. So what would have happened is in that germline cell, you're now going through replication. So this is where you replicate your DNA, which would be in the same phase. It would be an S phase of meiosis, S phase of interphase of a germ cell, of a germline cell. And now you've seen when they're replicated, how you've got 2 copies of every gene up and down the cell. So that will be your maternal X, In this case, I'm actually showing a male cell. So to be a male, you would have a Y. So this is a different germline cell. This is a male because now you have Y. And Ys always get passed from males to males to males. So in the next thing here is counting the chromosomes. So these chromosomes are once replication has happened. So DNA replication has happened. You've got 2 copies of every gene. And now what's very unique is once you go into the actual phases of meiosis. So meiosis is a reduction phase. So when we go through meiosis, we have prophase 1, and something very unique happens in prophase 1, and you can see in this picture is the parental chromosomes, known as homologous chromosomes. They pair up. So the long chromosome has found the long chromosome, the short chromosome has found the short chromosome, and in this case, the 2 X chromosomes are paired up. Next thing that happens is you have a process known as crossing over. So what you see is some of your maternal genes get passed on to the paternal chromatid, and some of the paternal go to the maternal. So it's an equal swap. So had we done this in class, we would have cut those little pieces out and we would have joined them on with tape. And then you can see how this is happening between your X's, between your long and your short chromosome. So you're swapping between what we call non-homologous sister chromatids. If you did a crossing over between sister chromatids, you would end up with the same thing. So you don't see any change. But we know that crossing over occurs and recombination. So this is the reason you do not look identical to your siblings. So prophase 1, 3 things are happening. You're having lining up of your homologous chromosomes, and then you're having crossing over between non-homologous sister chromatids. So this one and this one would be sister chromatids. This one and this one would be identical, but then you cross over between the maternal and the paternal chromosome. So color coding makes it nice and easy to see. Now, the next thing that happens is when you go into metaphase, notice that our homologous pairs are still paired up, and they're lining up in the middle. So instead of 46 individual chromosomes like we see in mitosis, they stay as homologous pairs. And this orientation can vary because I could have maybe had this chromosome on the other side, the orientation. So this also generates variation. So what's really cool, when you think of our chromosomes, we have 23 pairs. And when you go 2 to the power of 23, it ends up being a number that's a little bit higher than 8 million. So just by the random aligning up, if at metaphase 1 can generate about 8 million possible combinations. And that's why we are so unique, because we've rearranged, we're a rearrangement of our parents' genes before we were even formed. Next step is going into anaphase 1, and what you're seeing in anaphase 1 is the chromosome still consists of 2 sister chromatids as it's being pulled away. And you can see that how the variation is going with them to opposite sides. But we are now going, we're already halving our number of chromosomes, because if you look at our original cell, we have 6 chromosomes. 1, 2, 3, 4, 5, 6. Now we've halved the new cell that's going to be formed. Each new cell has only got 3 chromosomes. So 2 new daughter cells are formed with half the number of chromosomes. So we've only got 3. They're still replicated, but the cells are considered haploid, because haploid is when you halve your number of chromosomes. So for our cells, we have 46 chromosomes. After the end of telophase 1 in meiosis, each new cell has 23 chromosomes. Now, each of these cells is going to go a second cell division. They're going to move to telophase 1 and then go into prophase 2. But what's important to understand is there's no replication and no interphase. So you don't undergo another interphase. You basically just rest at the end of telophase 1. And now you see that every cell still has a chromosome of each type. We have an X, we have a short, and we have a long. There's our X, there's our short, and there's our long chromosome. So you have to make sure that your cells have one copy of every single chromosome. Now we undergo metaphase 2. So now you see the individual chromosomes lining up in one cell. Same thing happening in the other cell. And then you undergo an anaphase 2 in both cells. And now you see this is where those sister chromatids finally separate and move to opposite sides. So here's cell 1, here is cell 2. And at the end of this, we're going to form 2 new cells from cell 1 and 2 new cells from cell 2. So now again, these cells are haploid. So in the end, meiosis produces 4 daughter cells. Each one still has 3 chromosomes, but they are non-replicated. The cells are haploid. And when you look at them, you can see that they are non-identical. Because if you just look at just the coloring, you can see that the chromosomes are not the same. But what every single cell has to have is a complete set of genes on each of those chromosomes. So final question here that would have come in, these are called gametes. And because this was a female, these gametes would then be eggs. Had you started off with a male chromosomes, then you would have produced sperm. And then what we would have then done in class is a male and a female will have got together, and you would have randomly chosen one of your eggs or one of your sperm, and then we would have got together and made the offspring, which is kind of what we do in this lab. We make the offspring of the dragons. And what's really cool is this is where you get to do the genetics, when you actually start looking at how the genes work together, how dominant and recessive works. So that's where the little dragon one will come in and show you. So let me just go back off my screen. So this is the thing that you need to understand, is when you look at how your dragon traits are going to occur is going to be based on a little bit of understanding of genetics. So a gene is represented by a letter, and a capital letter means it is maybe usually the dominant effect. And a lowercase letter, you have to have 2 lowercase letters before you would actually have that effect. So, for instance, if we look at horns, horns are represented by a capital H or a lowercase h. And you can have multiple combinations of those alleles. But in order to have horns, you'd have to have at least 1 H. If you did not have horns, then you would need to have a little h from both of your parents. So if you look back at when you look at the actual chromosomes, in order to have horns, you must, the egg or the sperm must be contributing a big H. Same thing for wings. Legs are represented with L's. Whether you can breathe fire is an F. And then different color combinations are going to be combinations of A's and B's, and then whether you have a tail or not. So it's actually a really cool little simulation, because what you're going to do, let's just run through one quickly together. Hopefully it will work and you can see what you'll do. But everyone's will be unique. And then you'll be making babies and then filling out what your offspring look like. So I need to plug my laptop in before it goes dead here. To find Concordia University. Okay, so here's the activity that you get to do. So you're going to make some baby dragons. Before you do it, the best thing you can do to not get frustrated is read the directions clearly. So you will see on your sheet we have places where you actually fill out answers. And it looks like what's happened is it's defaulted to where I already did the activity the other day. But here you can see how the simulation will run. So here's what the mom looks like. So she's a purple dragon. She doesn't have any wings. She doesn't have any horns. She's got 2 little front legs. And here's the father. And he is a male, obviously. He's got wings and he has horns. So what happens when you undergo meiosis? So what you would do is you'd click on play here. And you can see meiosis is occurring in the female. So she's going to produce some eggs. And if you wanted to speed it up, so it's actually showing the process of meiosis slowly for you to watch. And ultimately, each parent is going to produce 4 gametes. So there's the eggs. And then you can actually hover over them and see what traits are in each egg. So if we looked at this one, has no plates, it's got a fire breathing, its color A, gene is little a and little b. If we look at the other chromosome, there's the horns, no scales, and this chromosome is the wings. Let's quickly make the sperm of the dad, and we will speed it up and get to the end. So there's his 4. And now what you do is you choose a gamete. So I'm going to choose that one from the female, and I'm going to choose that one from the male. And now I'm going to see what my baby looks like. Speed it up. Oh, it's a girl. And if we look at this girl, she's a purple female. She actually looks a lot like her mother. She has no horns and she has legs and she has scales on her back. So if you hover over the chromosomes, you can actually see the combinations that have actually occurred for her to look like that. So you can actually pull the chromosomes apart. Now, one of the things you'll also recognize is what chromosome did you need to get to be a female? And it's not telling you, but one of these chromosomes happens to be an X chromosome. Now, even though she's got the fire gene here, she's not breathing fire. So that'll be something else you'll get to watch. Once you've done with that, you take a snapshot and it will put it in here. And then these are going to be the pictures that you will upload into your report. And there's a couple of simulations you're going to do. And this one here is going to ask you if you can create an offspring with no horns. So that's where you're going to be a little bit more alert into which gametes you want to choose in the cross. And then the last question here is about, can you make a baby with wings or no wings? So follow through that, and then in the report that you upload, there will be these questions you will be answering, all kind of what you'll be uploading. So take time to do it properly. I did the activity. It did not take too long to do, but it was actually kind of fun based on what I've been explaining to you. But again, before you do anything, read the information, showing you how you're collecting your data, what you're putting it in, and what you're going to be getting. And then you're going to be asked questions, well, what phase of meiosis is this? So when we look at meiosis is a reduction division. It's not a cycle, because you start off with a diploid cell and then you produce haploid gametes that don't recycle back to the original cell. Those gametes go on and fuse and form a new individual. Then the last part of your meiosis lab deals with human genetics. So you're going to click on the Rutgers lab here, and you're actually going to go and do some karyotyping with chromosomes, and ultimately do some count chromosomes and see what happens when you have extra chromosomes. So this is a nice little activity, how we do have individuals in the population who have extra X's or extra Y's, or maybe they have an extra chromosome 21, and you'll get to read a little bit about that and then answer the last couple of questions. And then when you're finished with that, once you've done all of that and you've uploaded it, you will then, there's a post-lab quiz. So there's one for mitosis, and there's also one for meiosis. So even though it's only worth 7.5 points, there's not just 3 or 4 questions for you to do. But these are really great review activities to help you understand these processes. And again, the reason we do this is to help you learn your lecture material. So what I'd like you to do before we end the meeting is, I want all of those who are still here to at least go and say goodbye at the end, so that way I record in the attendance of the lab. And what I'll also do is this, I will post this, all these activities that I've already done, I will post that into the announcement as a Zoom meeting for you to go back if you want to use it to review in any way. So before we leave,